Hello everybody. Now we are discussing about the downstream processing. The downstream processing, these are the steps which are involved within the extraction and purification of the fermentation product from the fermentation broth. These are considered to be the downstream processing. Now majorly the two types of the products, these are formed through the fermentation process. Either these are the intracellular product or the extracellular product by using the microbial cultures. Now the extracellular products, these are the products which are synthesized by the microbial cells and then secreted outside the cell. These are considered to be the extracellular. And the intracellular products, these are the products which are synthesized by the microbial cells within the cells. Now the steps within the downstream processing, these are similar for both intracellular and extracellular products. One difference it is, in the case of intracellular product, one an essential step within the downstream processing, it is an extra, that is for the release of the intracellular product. This initial and extra step, it is involved within the purification, extraction and purification of these intracellular products. In the previous video, we are already discussing about the first step within the extraction of the extracellular product, that is the cell harvesting. So today we are focusing on for the extraction and purification of intracellular product, which is the initial step. That is, this initial step, it is nothing but the release of intracellular product. Now there are the various types of the product which are synthesized inside the microbial cells. Either they may be vitamins or the enzymes. These are first, and majorly these processes or these products, as these are synthesized within the cell for their extraction and purification, firstly initially it is important to release this intracellular product from the cells. Now for that purpose, the microorganisms or the other cells which are synthesized, these are firstly get disintegrated or disrupted by either the physical, chemical or the enzymatic methods. Now for the selection of either physical, chemical or any enzymatic method, it is totally depending upon from which type of the cells we want to extract that fermentation product or release that fermentation product, and how there is the product which we want to separate out, what is the characteristics of that product. Now for example, the gram-negative bacteria and the filamentous fungi can be easily broken down as compared to the gram-positive bacteria or the yeast. Now here from this diagram we can understand which types of the methods these are involved within the cell disruption. Now majorly the three different types of the methodologies these are applied for the cell disruption purpose: the physical method, chemical method and the enzymatic method. In the physical method, there are the 6 different steps these are involved. That is, it is an ultrasonication, osmotic shock, heat shock or thermolysis, high pressure homogenization, impingement method and grinding with the glass beads. All of these are the physical methods. Now the chemical methods. Chemical method, there are the 3 types. That is the alkaline, use of alkaline treatment, organic solvents and the detergents. Third one, it is the enzymatic method. The common enzymatic method, it is the use of the lysozymes. Now firstly we are focusing on the physical methods for the cell disruption. Now the microorganisms or the microbial cells, this can be disrupted by using certain physical method for the release of the intracellular product. Now the first method, it is the ultrasonication. Now this ultrasonication, it is a widely employed method at the laboratory scale due to its high cost, and it is not suitable for the large scale use within the industries. Now within this ultrasonication, the sound waves, these are, that is high intensity sound waves, these are utilized for making the vibration within the microbial cells, which has resulted within the disruption of the microbial cells to release the intracellular product. Now the next one, it is the osmotic shock. Now within the osmotic shock, here the cells, these are placed within the high solute concentration. This method involves the suspension of the cell, that is free from the growth medium, only the microbial cells, in the 20% buffered sucrose. Means here within this we can understand that the cell disruption that we are discussed within the earlier video, this the cell harvesting process that we are discussed within the earlier video, firstly it is for that is the separation of microbial biomass, and after that we are applying this cell disruption process for releasing this intracellular product. So the cell biomass that we are obtained from the cell harvesting process, Majorly it is treated or it is kept within the 20% buffered sucrose solution. The cells these are then transferred to the water at about 4 degrees centigrade. This osmotic shock it is utilized for the release of specifically the hydrolytic enzyme and binding proteins from the gram negative bacteria. Now the next one it is the heat shock or the thermolysis. Now the name itself indicates that is the principle of this process. That is within this the breakage of the microbial cell or the disruption of the microbial cell it is carried out by application of the higher temperature. Now the heat it is relatively easy and cheap, means the cost it is get lower whenever we are applying this process. But the most important limitation of this process it is only used only for a very few heat stable intracellular product, because within this process we are for the cell disruption purpose we are applying the higher temperature. So most of the enzymes or proteins these are heat labile in nature. So those of the intracellular products which are showing the heat stability, only for these products we can apply this method. Now the next it is the high pressure homogenization. Now this technique it is involves the forcing of the cell suspension at the high pressure through a very narrow orifice to come out the atmospheric pressure. Means there is the transfer of the cells with the high pressure from a small orifice, and this sudden release of the high pressure creates a liquid shear that can be break the microbial cells. Means due to the application of pressure, the pressure it is created on the microbial cells, due to which there is the disruption of the cells. The next one it is the impigmentment method. In this procedure, the stream of suspended cells at the higher velocity and the pressure, these are forced to hit either stationary surface or a second stream of suspended cell. Means there is the collision of the microbial cells. There is collision of microbial cells. The cells are disrupted by forces created at the point of contact. The microfluidizer it is a device which is developed to based on the principle of impingement method, and the advantage of this technique it can be effectively used for disrupting the cells which are at the low concentration. Then the grinding with the glass beads, it is a last physical method applied for the cell disruption. In this method, the cells these are mixed with the glass beads and then subjected to the high speed in the reaction vessel. Now there is the breakage of the cells as they are forced against the wall of the vessel by the beads. Now there are different factors which can influence the cell breakage, that is the size and quantity of the glass beads, concentration and the age of the cells, the temperature and the agitator speed. Under the optimal condition, one can expect a maximal breakage of about 80% of the cells. Means it is an efficient method for the disruption of the cells. Now we will switch over to the chemical methods for the cell disruption. Now within this there are the three techniques, that is the use of alkalis, organic solvent and the detergent. Now whenever there is the alkali treatment, this alkali treatment can be used for the extraction of some bacterial proteins. Now whenever we are utilizing this technique, one point it is taken into consideration that this method can be applied only to the extraction of those products or the cell disruption of microbial cells whenever we are extracting those of the intracellular products which are having the alkali stability, which can stable at the alkaline pH. For example, the recombinant growth hormone which is produced within the E. coli cells, it can be efficiently released from the E. coli cell by the treatment with sodium hydroxide at the pH 11. Now the next method it is the organic solvent. Now there are the several water miscible organic solvent which can be utilized for the purpose of the cell disruption. The examples of some organic solvent includes the methanol, ethanol, isopropanol and butanol. Now as these organic solvents these are inflammable, hence they require the specialized equipment for the fire safety. The organic solvent toluene it is frequently utilized, and it is believed that the toluene dissolves the membrane phospholipids and creates a membrane pores for the release of intracellular product, which is beneficial from the gram negative bacterial cells. The next one it is the use of detergents. Now these detergents these are the ionic in nature, that is cationic cetyl trimethyl ammonium bromide or anionic sodium lauryl sulfate. This can be denatures the membrane proteins and lyse the cells. Means the main activity of these quaternary ammonium salts or these detergents, it is to denature the membrane proteins, and whenever there is a denaturation of membrane proteins, the cells these are get lysed. The non-ionic detergents, these are less reactive than the ionic ones, and this can be also used for the some extent, that is the Tetron X100 or the Twin 80. The problem with the use of detergents, it is they are affect the purification steps, particularly the salt precipitation. So this limitation can be overcome by using the ultrafiltration or the ion exchange chromatography for the purification process. The next one, it is the enzymatic methods for the cell disruption. Now the cell disruption, it is carried out by using the different enzymes. These enzymes, these are responsible for lysing the cells. The lysis of the cells occurred under the mild condition in a selective manner. The most important enzyme which is utilized for the cell disruption, it is the lysozyme. It is commercially available, produced from the hen egg white, that is it hydrolyzes the beta 1,4 glycosidic bonds in the mucopeptide of the bacterial cell walls. The gram-positive bacteria with the higher content of the cell wall mucopeptide or the peptidoglycan layer are more susceptible for the action of lysozyme. And for the gram-negative bacteria, as we know that within the gram-negative bacteria, the outermost lipid bilayer it is present. So for this lipid bilayer dissolution, these lysozymes, these are used in association with the EDTA, which can break the gram-negative cells. As the cell wall these are get digested with the lysozyme, the osmotic effects break the periplasmic membrane for the release of intracellular content. Certain other enzymes can also be used, although these are less frequently used for the cell disruption. For the lysis of each cell walls, there is the utilization of the glucanase and the mannase in combination with the protease enzyme. It is depending upon the cell wall composition of the microbial cells. So these are the different methods which can be utilized for the disruption of the cells for the purpose of release of the intracellular product. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.